Okay, so the average of the test was good. It wasn't as bad as you made me feel you did. So, well, you made me feel by saying it was hard, tough. So I thought the results will be poor, but actually it was good. 76 is the average that we've seen in the past years. It was odd last exam that the average was 90 or whatever. This is a normal average. So those of you that are at average or higher, that means you did really well. Others, there were not many under this average. Few, but not a lot. So it was good. And I will post the key. I don't know if I did already, but do please look at the key. It's helpful. All of the material will be part of the final exam. So if you make sure that you look at your answers and how they compare to the key, that will be helpful. If you have questions concerning the grade or the grading, please talk to me directly. Chelsea goes through grading, and then I look at each paper myself as well. And between the both of us, I think your grade is fair, but let me know if you have any questions. Okay, just finishing up last few slides of minerals. What are the different categories of mineral analysis methods? We have 3 different categories for mineral analysis methods. What were they? Remember? What lab did we have earlier in the semester? AA? Other than the AA? Huh? Lucy? Colorimetric assay. So we can do atomic absorption or ICP, or we can do colorimetric, or we talked about one other method, type of methods. Peter? Titration. Complexometric titration method. Okay. What are some considerations we talked about in sample preparation and analysis? If you don't remember logically, what would you consider when you're preparing samples, Mallory? Contamination from? From the milling, yes. If it's volatile or not, that would be if we're doing ashing. I'm talking about just the preparation for mineral analysis, not necessarily preparing or ashing the sample. I wanted to go back to contamination. We run blanks often because not only we mill the sample and you might get contamination from the mill that you use, you might get contamination from reagents, from glassware, so you always have to run blank. We talked about the pH of the system, the temperature, the complexity of the sample, and how sometimes you need to prepare the standards in the same complex material that the sample is composed of. So why is it important to analyze minerals, Stephanie? Water quality is a big one. What else, Haley? Presence of heavy metal.
I was going to say processing effects.
Processing effects, such as the refining example of flour, where when people enrich the flour by the same amount of elements that were lost due to refining. What else, Justin? Nutrition label and adequate intake of minerals. Let's see. How do we determine hardness of water? There is a method we talked about last time. In hardness of water, what component that we usually measure? What mineral element we usually measure? Yeah? Calcium? Calcium. Yeah. Which method? It's a titration method by EDTA. It's a complex symmetric titration method. What does more titration measure? Remember? There's test strips of it as well. Salt? I answered, sorry. I wasn't thinking. I saw your hand and then I answered. Salt. And how is it different from Vollard? Both measure salt. One is a forward titration where you titrate directly with the reagent. And then the other one is you have an excess of the reagent and then you titrate the remaining of that reagent. So it's a backward titration. Okay, moving on, other than titration or titration methods, you have the colorimetric methods. An example of that is measuring phosphorus using molybdovanadate reagent, which is a yellow reagent. When the reagents reacts with the phosphorus, you get the yellow color. So the higher concentration of the phosphorus, the darker is your yellow color. It's a very beautiful set of standards. When you prepare the standards, the shades of yellow. But this method requires ashing. It definitely follows Beer's law, so you have calibration. So you would have standards and you do a standard curve, and that's the most common spectrometric method used for phosphorus. Last is your ion selective electrode. So it's like the pH meter, but the electrode would be sensitive to particular ion instead of hydrogen. So you would have, it measures the cations and anions. You can have electrodes specific for calcium, sodium, chloride, potassium, and fluoride. And then the difference in potential can be converted to concentration of the ion, and you can also do standard curves to get the exact concentration. That means you prepare standards of calcium or standard of sodium, you analyze it by the ion selective electrode, and then you put your sample and determine the concentration by using a calibration curve. Some consideration for that is that you might have other ions in the system. So you might have, other than sodium, you might have iron or zinc or magnesium. So you will have different ionic activity. So in order to get accurate results, your samples and standards should have similar ionic strength other than the actual element that you're measuring. Sometimes you have interfering ions. That means the electrode could be sensitive to other ions. That would lead to about 10% error. It's a very sensitive method, can read up to or as low as 10 to the negative 6 molar. And however, if the concentrations are really low, the disadvantage is that it takes longer to give a stable and reliable reading. It's like with the pH meter, if you remember, you had to put it in there when you were titrating, you had to wait a little bit for the number to stabilize. These are some examples of applications: salt and nitrite in processed meat, calcium in milk, salt in butter and cheese, potassium and sodium in wine. And we did solve this, and that's the end of mineral analysis. Any burning question? Okay. So, moving on to a new topic altogether. Only if I get my hand to work. Immunoassays. I should have Chelsea give this lecture.
I did it when you were on sabbatical that year.
You did it one time, that's right. You want to take the floor? Well, Chelsea did her master's on allergenicity of soy, so this is a very common subject for 2 years, 3.5 years.
Yeah.
Okay, so immunoassays, what do we know about them? What are immunoassays and what do they mean? Have you heard of immunoassays? No? Nobody heard of immunoassays before? I doubt it. No? There's an antigen in there. You're measuring an antigen.
Is that like ELISA?
Yes. It is, ELISA is an immunoassay. There are different forms of immunoassay. What does ELISA stand for? There you go. Did you use it before?
We did it in micro, right?
Yes. Yep.
Ah.
But we didn't really. I think there was. We like kind
We just kind of did it, yeah.
They made us sit for like an hour after.
They wouldn't tell us anything about it.
They won't tell you anything about
They just doing
No,
that.
actually they did it. It was Lorenko.
Okay.
That explains it all.
excitement about immunoassays. Calm down. All right. So immunoassay, by definition, you're using antibodies, basically. Antibodies, antigen interactions. And you can make use of this interaction for qualitative and quantitative identification and detection of a certain component. Okay? One component could be bacteria, because the cells on the membrane of the bacteria, or the cell wall of the bacteria, on the cell walls of the bacteria, you have proteins or antigens that are recognized by antibodies. And the antibodies would be labeled one way or the other by enzyme or color or by any indicative measure that you can quantify. So in immunoassays, you are basically relying on a very specific interaction between an antigen and an antibody. And like I said, there are different forms of it. ELISA is one form. Another form is Western blot is a form. And immuno affinity chromatography is another form of it. Okay. So just a quick overview of the concept and theory. I'm not going to dive too much into that. But antibody, I'm sure you've learned about antibodies somewhere in biology along the way. So antibody is basically 4 peptides or polypeptides. 2 are long chain or heavy chain, and then 2 of them are light chain. So the 2 light chain and the 2 heavy chain, they're identical. Okay? And then the most important component of the antibody is the binding site. So the binding site is where the antibody binds very specifically to an antigen in a location at the antigen. That location we call epitope. Epitope is the region on an antigen that binds to an antibody. So how antibodies are produced very quickly in the body? Once a foreign body is introduced, antibodies are formed by B cells. Of course, there's a lot of mechanism that goes into that, but B cells respond to an antigen and produces antibodies. And these antibodies become strongly affiliated or have strong affinity to that antigen. So the affinity is really the strongest non-covalent interaction between any 2 molecules. So the affinity is so high that the affinity constant is 10 to the 12 moles per liter. The interactions are non-covalent, so they're the ionic, the hydrogen bonding, the hydrophobic, and the van der Waals. All non-covalent bonding or interactions can take place when the antigen is binding to the antibody. So like I said, the most important part of the antibody is the binding site. And it is usually a small, relatively small site. and equivalent to the size of 7 glucose units. So it's a small, really small site. And like I said, the antibody binds to the antigen via a very specific region, and that's what we call epitope. There are mainly two types of epitopes. If it's a protein, mind you, if it's a protein, if the antigen is a protein, you have linear epitope where you have a sequence of amino acid, 1, 2, 3, 4, 5 in a sequence, and that would be a region where the antibody would bind. If it is a conformational epitope, you know, a protein folds. So the folding would make the 1, 2, 3 on this side and the 4, 5 on the other side of the protein. But due to the folding of the protein, they come together and form an epitope. We call that conformational epitope. So another term that you will hear and will be relevant to different types of assays is polyclonal and monoclonal antibodies. So antibodies, they come different types of antibodies. Even for the same antigen, you will have multiple, you can have multiple types of antibodies. So the B cells, each B cell will produce one type of an antibody that will be specific to one epitope. Okay? We call this monoclonal. That means a mixture of antibodies, all of them are react with one type of epitope. I'll give you an example where monoclonal is important to have in an assay. Polyclonal is when multiple B cells produce different antibodies. Each antibody would have specificity to a different epitope. So say, let me get this, and can you pull that out there? Thank you. All right, so you have the protein, let's say, is your antigen. Of course, there are different types of antigens, but if we say the protein is your antigen, then you will have, let's say, here's one epitope, here's another, here's another. So you will have different antibodies that can bind to the same antigen but at different locations. These are polyclonal antibodies. Okay. So now I'm just giving you terminology, and then you'll hear me come back to them later. Okay. Obviously, this is just for your knowledge. Obviously, when an antigen is injected in a body of an animal, let's say, to generate antibodies, usually the body would produce a polyclonal antibodies. So in order to get monoclonal, you can either do chromatography to separate a certain type of antibodies, or they have found a way to get cancerous cell and link it to B cells, one type of B cells, and have it, you know, divide and increase in number and produce one type of epitope, or one type of antibody, and then you can have a monoclonal antibodies that can be used in standard assays. Okay, so the body obviously will not only produce one type of antibody. So characteristics of the immunoassays, yes, Stephanie. Yeah. Yeah. So, you know, the protein is not a linear sequence. I mean, it's a linear sequence, but it is not, it doesn't stay in its primary form. It's going to fold. All right? And where it folds, 3 amino acids from here and 3 amino acids from here can form one epitope. We call it conformation because it's due to the conformation of the protein. Whereas if the epitope binds here, you don't have folding and you just have amino acids in sequence, that would be linear. I don't know if you can see from the back. Okay. So back to this, immunoassays, in order to be used in efficient manner in analysis, they have to be very specific so that we are measuring a specific antigen. Sensitive, so we can detect at low concentration, especially if we're after allergens, for example. We need assays that would detect at very low concentration because you don't want to have a food product that's contaminated with allergy, with an allergenic component and not label it. So it's very important to be sensitive. And simple, we love simple. So simple assays are always good. We need to find an appropriate antibody. Sometimes you have it available as a kit, so you can buy a kit to determine, let's say, presence of horse meat in beef patty. It's a thing, you know. I think there was a case about that. Having horse meat in a beef patty. You've heard about it? You didn't hear about it? Where did it happen? It happened in Europe? Yeah. So you can buy specific assays to determine a foreign body that shouldn't be where in that product. So we're not only we want to find the antibodies. So like I said, you can find them as kits. You can actually, like in Chelsea's thesis, what we did is we get human blood from people that were allergic to soy, and that was the source of our antibodies for the assays. So we need to have a source of antibodies for our assays. We need to be able to separate and differentiate. So if we have different proteins in there, I have the milk protein and I have the soy protein and I have the peanut protein. Well, that's a bad example. Peanut and soy, there's cross-reactivity. All right, meat protein, beef specifically, and they're all in a mixture. And you want to see if there's a contamination with soy. Then you need a specific assay that will pick up only soy. Okay. A common feature that we rely on is that proteins. Antibodies are proteins, right? So proteins, whether it's the antibody or the antigen, we rely on their hydrophobic properties to bind to hydrophobic surface, because the first part of the assay is to bind either the antigen or the antibody to a hydrophobic surface. Have you seen a microplate before? We do the ELISA assay, for example, in microplate. Did you do it in microbiology in microplate as well? Tiny little wells. We like to keep them together in solidarity. Anyway, so this is a plastic, so it will have a hydrophobic surface. So the first thing that we have in there, the first solution will either be an antibody or the antigen, protein antigen. Then they will bind to the bottom of the well by hydrophobic interaction. That's key. We need them to bind, because we want to assess their presence. If they don't bind, we can't quantify the antigen. If the antibody cannot bind or the antigen cannot bind, we cannot continue with our quantification. So that is very essential. We rely on the hydrophobic properties that allow us to link them to a hydrophobic surface. How do we produce antibodies specific for measuring a target antigen? I think I said something about that earlier. Yeah.
So we can use chromatography to separate them?
Produce them before separating them. How do I get them? Of course, they are biological, so they have to come from a body. So usually, if we want antibody, there are a lot of antibodies from goats, for example. So what they do is they inject the animal directly in their bloodstream with the antigen of interest. And then the animal will produce antibody, will collect the blood, and then isolate the antibodies from the blood. And another way I told you about to produce monoclonal is the use of cancerous cells and linking them to B cells and producing, in a cell culture, producing monoclonal antibody. Okay, so what are some applications? We talked about bacteria. Pregnancy test, yes. They use specifically flow strip, and I have it in my slides. Did you see it in my slides? Okay, but that's true. Stephanie? Aflatoxins, toxins presence. Julianne?
I was going to say something about allergens.
Allergens, yes. So toxic matter, allergens, presence of different protein species. The pregnancy test that Emily is talking about is a specific protein in the urine. And we said aflatoxin as an example, identification of bacteria and viruses, and genetically modified organisms. So there are others, like antibiotics, for example. So there are other applications that we will talk about and examples that we'll talk about. But these are some of the main ones. Yeah, this meat species authentication is the example I gave you, that there was a case where they suspected horse meat in a beef patty, and they found that by running immunoassays, by getting an antibody specific for horse. Okay. So the different types, these are examples of different types of immunoassays. So one of the very common ones that used a lot is called the sandwich ELISA. And you basically make a sandwich of the antigen. And your toast pieces is the antibodies. So it's a very common, and ELISA stands for Enzyme-Linked Immunosorbent Assay. Sorbent is because of linking, adsorbing to the hydrophobic surface. There is the comparative form of ELISA, and there is an indirect form of ELISA, and we'll talk about each one of those. Other immunoassays, the immunoaffinity purification, either purification of an antigen or purification if you want to produce a monoclonal antibodies. The lateral flow strip, which can be used as a field test. So if you want to determine presence of GMO on a field of soybeans that they claim it's organic non-GMO, so you don't have the facilities or the lab that can allow you to do tests, wet chemistry stuff, you can do a lateral flow strip, and we'll talk about that. The pregnancy test is a lateral flow strip. And then Western blot is a qualitative means of detection the presence of the antigens, so you can detect if they're present in a mixture of proteins, which one of these proteins react with or are allergenic. If you are looking at a mixture of proteins and trying to determine which one of these will react in a body of somebody that has allergies to soy or to peanut or to milk protein. So Western blot can be used as a qualitative measure. We'll talk about how it works. So starting with enzyme-linked immunosorbent assay, from the name enzyme-linked, you link an enzyme, and then you use a substrate that will turn into a product that has a color that you can detect. So it's a means of quantification. Originally, before enzyme-linked, it was the radioactive iodine that they would label, but they found that there's another easier way of detection is using enzyme. Okay, so the common enzymes are most common is the horseradish peroxidase, and another one that is common is alkaline phosphatase. They have quick reaction rates. You can get identification and detection pretty efficiently. And they are easily linked, can be easily linked to an protein antigen or the antibody. So you get, I said that they're stable, they're easily linked, rapid, and then we talked about the substrate conversion to a product. And we need an ELISA plate reader. So basically when you run an assay, how many wells are there in here? 96 wells? Yeah, so you can really run 96 samples in one shot. So microplate reader, I'll show you a picture of it. So once you prepare your reaction mixture, you would just put it in spectrophotometer like you had in the lab, but instead of reading an absorption of each tube one at a time, you will have 96 readings simultaneously. So it's very efficient to have a standard curve. Yes?
I'm a little confused about how this works because the enzymes are separate from the antibodies and the antigen, right?
The enzymes are linked, and I'll show you. They link them via covalent interaction. They link an enzyme to antibody, or they link an enzyme to an antigen.
So once the antibody and the antigen
React, bind,
then the enzyme produces something.
then you add a substrate, and the enzyme convert that substrate. And I'll talk about all these steps.
Okay.
I will not leave you like that. I will talk about all of the steps in a minute here. So that's the plate reader the plate is going around for you to see. So you will prepare your standard curve and the samples, and then that's a spectrophotometric analysis. So you have a spectrophotometer and then a place where we call it the plate holder. You put the plate and it goes in, and oftentimes, if not always, you have a controlled temperature because you want the reaction to occur at a certain temperature, and then after an increment of time, you get a measurement. And then you get simultaneous measurements of all the wells. So it's a pretty efficient way of doing this analysis. So now main steps that Mallory was curious about. I'm going to explain the main steps in terms of sandwich ELISA. Okay. It's going to take a while. Okay. All right. So let's say this is the surface of one of the wells, one of the 96 wells, magnified. So the first thing that you would add, I'm explaining this as a sandwich ELISA. So if it's a sandwich ELISA, the first reagent that you will put in is a solution of your antibodies. So your antibodies will bind. You would hope they bind face up, but you cannot control how they bind. So here's your binding site. No, here there's no binding site. Here are your binding site, and then since they're proteins, so they're going to bind to your surface via hydrophobic interactions. Okay. Then there is a wash cycle. We have a plate washer. You put, what do you put to wash?
I use the PBS. So
Buffer. So you put a buffer and you wash it, so any non-bound antibodies would be removed. Okay? So the plate will only have bound antibodies. Now there are still surfaces here, right? You don't want that surface to be available to your protein antigen. Let's say you're doing soy protein. That's the easiest example. You don't want soy protein, if you want to detect soy protein, you do not want the soy protein to bind here. You want it to bind to your antibodies. Soy protein has hydrophobic surface. It will potentially bind here. Then you won't have a detection. You want it to bind to that. Then you add a blocking agent, block the remaining surface. So the next thing you add is a blocking agent. The blocking agent is a protein usually, so we use bovine serum albumin of a specific quality that will block all possible binding sites, remaining binding sites. And then another wash step to remove any excess bovine serum albumin. Then you add your sample. A solution of your sample. If the sample has soy protein, what will happen? It will bind. So you will get, no, not here. You will get soy protein bound. All right, then another wash step to wash everything that didn't get all the other sample components. You don't want anything in there.
What was that stuff that
The what?
just did?
This?
Yeah.
That's when you put the sample.
Oh,
If you have soy protein, it will bind. I'm just giving an example of soy protein, assuming that's the antigen. It will bind. Everything else will be washed off. Then, since it's a sandwich ELISA, you will have another antibody. It would be great if the antibody, not lambda, antibody, if the antibody that you add here, it would be great if it's monoclonal. Then you will have very specific, only one type of antibody binding. Monoclonal labeled with an enzyme. It would be great thing to already purchase something that's already labeled. So now you have your sandwich. It's your antigen, antibody, antibody. This antibody is labeled. You do another washing step. It's always there's a washing step. Every time you add the reagent, after you allow it to incubate for a certain amount of time, you wash, and then now you would add your substrate. Then you add substrate. The substrate will react with the enzyme, will give you a product, and then you will measure absorption. So
The enzyme attaches on
you can purchase already a labeled antibody,
with the enzyme.
labeled with the enzyme. Yeah. So already, you can do it in the lab. It's just longer process. But if you can get an antibody already labeled, that would be great. Yeah?
So, if the substrate, so if the first antibody doesn't bind to the protein because there isn't enough protein in the sample,
Then you don't get absorption.
so the substrate only reacts if like that whole thing is reacting.
Yes, because you have a washing step. So if you don't have an antigen, the next antibody will not bind. And when you wash it, it will wash off the antibody that is labeled with the enzyme. So if you don't have an antigen here, if you don't have an antigen, when you add this, it won't bind with anything. You wash, it will wash off. And then when you add the substrate, there's no enzyme. So you don't see an absorption. So that's why this can be, you can do standard curve and you can quantify. Because the concentration of the antigen, the higher the antigen, the higher the binding, the more you're going to have substrate changes to product. You will have higher absorption. Okay, so in sandwich ELISA, the higher the absorption, the higher the antigen presence. Okay, the washing step is very critical because if you don't have binding, you're going to wash everything off.
So we want excessive amount of the first antibody.
Yeah, well, they're not necessarily excessive, enough to make sure that you have enough bound to the plate. So this method would be established. You have established methods for that. Yes, Bailey?
The amount of antibodies that bind to the substrate is what you measured.
What, say that again.
Because it's like the substrate binds to the, is it the antigen initially?
The,
What is it for?
so the substrate, the substrate that we add here, so this is an enzyme, right? So your antibody is linked to an enzyme, hence the name enzyme-linked. So the antibody has an enzyme When you add the substrate, it's specific to that enzyme. So the enzyme is found now in your well, and it has a substrate. It will convert it to a product, then has color, and you measure absorption. Stephanie? No, no. You always have this happen, so that's why your experiment with the reagents and the concentration is all being figured out, so you would get enough sensitivity. Yeah. No, you already blocked. Okay, so you establish your method basically. You always establish your method to have enough antibodies that would bind. So this has been figured out for you with the different methods that are available. Or you spend 2 years figuring it out. Yeah. It's not. Any other questions? That's exactly the drawing here for a sandwich ELISA. So that's the illustration that I've illustrated right now and talked about. Now, this is what we call a direct ELISA. So you are directly binding and directly measuring. So there is another form we call indirect ELISA. So the indirect ELISA, now you're going to say, what? Okay. But we'll let Sophia leave, and Sarah will tell her the what? Okay. So indirect ELISA, this is another sandwich ELISA. Okay, so here's your plate. That's an animation. So here's your plate. Here's your capturing antibody. Oh, did the other figure have label? Hold on, let me go back. Yeah. So I want you to know that this is called capture antibody. The first set is capture. It's capturing the antigen. Just in case you get a figure and ask you to label, you need to know this. Okay, capture antibody. This is your target analyte or antigen. The solid phase, that means this is your hydrophobic surface. The detection antibody is the antibody that is labeled with the enzyme. And then you have a substrate and you have a product. So you want to know the different components, huh? Oh, the horseradish enzyme. There are two different types. It's on one of the slides. Or they can have alkaline phosphatase. So usually there are common enzymes. Horseradish peroxidase or alkaline phosphatase. These are the most common enzymes used.
Is there a reason why it's called horseradish?
Comes from horseradish. Yeah, I would say that too. Okay, going to indirect ELISA. So it's the same thing, but listen to this. This is very important. So you have your capture and you have the other antibody that will bind to your antigen, but this second antibody is not the detection antibody because it's not labeled. You have an antibody to the antibody. Now you have to tell me what? What? So antibody to the antibody. So let's say in Chelsea's case, that's the best example, we use human sera to do the soy protein assay. So she will put sera first. The antibodies from the sera will bind. Sera, that means the processed blood. You centrifuge, you get the serum. And in the serum, you have the antibodies. So the antibodies will bind, and then your antigen, if present in the sample, will bind. And then she will, after washing, she will put another serum sample. So antibodies will bind to this. Instead of having her try to label an antibody, just get an enzyme and initiate an interaction, which is a tedious procedure. So to prevent from doing that and to enhance sensitivity, we purchase antibody to human antibody. Antibody to human antibody. Because antibodies are proteins. So these has been, do you use goat antibodies? Goat?
I think it was
Goat.
or was it anti-rabbit?
Anti-something, rabbit. So if we use a rabbit, it would be antibodies from rabbit that are antibodies to the human antibodies. Human antibodies are proteins, so they get injected into the rabbit or goat, and then the goat or the rabbit will produce antibodies to these antibodies. These we can purchase. They are labeled already and biotilated. I'm not going to go there, biotin and evident. But anyway, to enhance the sensitivity of the detection. You have multiple regions on the antibody where it has multiple enzymes to enhance the sensitivity, and then the substrate is added, and then the substrate gets converted to a product, and voila.
Why is it such an increase in sensitivity?
Okay, you want to know. All right, so you get a butylated.
Well, isn't that like why you would do this one over the other one?
Yeah. Well, yeah. I mean, yes. I didn't want to go into details, but I will. In the last minute. So you have biotin. Biotin is a vitamin, right? And biotin and evidin is a protein, has very high selectivity to binding to biotin. So you can have several biotins attached to the antibody. And then you will have evidin or streptavidin in this case. Chelsea, correct me, leave your phone. Yeah, okay. Evidin bound to biotin. And then the evidin is labeled by the enzyme at multiple locations. So you will have multiple enzymes on the evidin, on the protein. No, that's the evidin. Okay, I'm going to draw this. And that's biotin.
I was trying to understand it more, but I feel like I just confuse everybody else more. So.
And then enzyme. Can I finish, Chelsea? Sorry. So you have biotin attached to the antibody, and then the evidin is a protein that's very selective to binding to biotin, and you have multiple locations on that protein that you can have enzyme linked. So one or potentially two antigens sandwiched will get you very high sensitivity because you have multiple substrate reacting.
Right, so you're basically just increasing
Sensitive.
the amount of substrate that can react.
Yes, so you're increasing the absorption. Yeah, with only a little bit of antigen, you're having a compound that have multiple enzymes. Do you need to add?
Right. And when you think of each layer, in an ELISA, things can happen. So if you're detecting by adding this, what we would call like a secondary antibody, so you have a detection antibody, and then the one that Pam's talking about here is the secondary antibody, it means that we're less likely to get false negatives. So our absorbance for what it is less likely to be that something bound elsewhere. So it's increasing that sensitivity of what we're detecting.
Yeah, and then the other thing is, yes, it's called secondary antibody. These are primary antibodies. They're called primary because they react directly with your antigen. The secondary, it reacts with the antibody. Okay, thank you.